Good morning for everybody. I'm Beridze Raymond Anzorovich, the pupil of the on the MAC Young Researcher KPNZ Odessa Ecological and Naturalistic Sense Afalina. I want to present to you my scientific research. Development of effective methods of propagation of berry yew and virgin juniper in the conditions of Odessa and the prospects of creating preparations with herbicidal and stimulating action from extracts of conifers. Military actions on the territory of Ukraine led to the destruction or violation of forest belts, parks, forest massifs, and protected areas. Therefore, the need for high quality and cheap planting material of valuable tree species is more urgent than ever. Valuable mother material for seed propagation of these beautiful exotic trees grown in the park monument of horticulture art Humanitarian of the regional Humanitarian Center. Also, these plants can then be used to create drugs with herbicidal and stimulating effects. Therefore, the aim of our work was to study the methods of mass propagation of blueberry and virgin juniper and to study the effect of physiologically active substance of extracts of blueberry, virgin juniper, Himalayan pine, black pine, cedar of Lebanon, and cedar of Atlas in different concentrations and under different conditions to create physiologically active drugs. The task of the research was literature search on the topic of research, collection of berry yew seeds and their preparation for sowing by the methods of certification, developers of methods of propagation of berry yews and virgin juniper under mother plants, study of methods of vegetative propagation of yew, analysis of methods of mass propagation of berry yew and virgin juniper, and recommendation for practical use of research results. Conducting a series of spring, summer, autumn, and winter experiments on the analysis of influence of coniferous plant extracts on the growth and development of fruits of reddish berries, national and saxa, and soft wet berries, Nауки and Luzanivska. Study of the effects of coniferous plant extracts in vivo, analysis of the obtained data, and their application for the development of drugs with herbicidal and stimulating action. The object of the research was a berry yew, virgin juniper, black pine, Himalayan pine, cedar of Lebanon, cedar of Atlas. We also used soft wet of the Arnautka and Luzanivska varieties, radishes of the Saxe and national varieties as an object of research. The work was carried out in the Yunatsky Park monuments of garden and park art of the regional Humanitarian Center and the Odessa Ecological and Naturalistic Center Afalina during 2020 and 2024. To grow berry yew and virgin juniper, we use methods of seed propagation, vegetative propagation, and the methods of growing yew and juniper seeds under mother plants. To study the action of coniferous plants extracts, we used the biotesting methods. In our work, we also use standard mathematical methods to analyze research results. Results of the seeds methods of berry yew propagation. The seed methods using certification did not produce a single seedling. We associate this with the fact that we exempt only 1,000 seeds, as well as the fact that certification methods are not sufficient to start the process of seed germination in order to destroy the strong shell of berry yew. It is necessary to apply chemical and mechanical means of seed scarification and to provide seeds with constant moisture. The vegetative method gave results only the second time. The previous time when we did the experiment for the first time, it did not produce a single seedling. The time we grew 3 cutting from 10 cutting. Therefore, we consider that this method is effective enough for the propagation of berry yew. The results of the methods of growing berry yew seeds in the soil from under the mother plant. Over 4 years, we have grown more than 1,000 berry yew seedlings under one mother plant. In 2021, according to our methods, to the first time we received a large number of virgin juniper seedlings under the mother plant. According to our calculation, 350 seedlings sprouted from 10,000 juniper seeds, which is 3.5% of seed germination. Work on the study of effects of coniferous plant extracts began in March 2022. For 2 years, we studied a lot of literature and performed more than 200 experiments. A number of factors influence the effect of physiologically active substance of conifers on wet and reddish seeds. One of these factors is the use of leaves of different ages. In spring experiments, we used all leaves, and in summer experiments, we used young leaves of Beridze and Himalayan pine and other conifer plants, but only a different stage of development. The second factor that influenced the results of experiments was seasonal fluctuation in the context of physiologically active substances like terpenes, phenols, and other. The third factor, or rather a set of biotic factors such as insulation, precipitation, and temperature. The fourth factor that influenced the different results of the experiments was different concentration of the extracts. Small concentrations such as 10 gram per 250 milliliter or 1 to 10 showed either a weak inhibitory effect or were equal to the control, and in many cases stimulated the growth of plants compared to the control. An interesting physiological effect was observed with extracts of cedar of Lebanon and Atlas in concentration of 10 grams of leaves per 250 milliliter of water. In all experiments, we recorded a strong stimulation of seed germination on the extracts of cedar of Lebanon and Atlas cedar. The most successful method of berry propagation turned out to be the method of growing seedlings under mother plants. It allows growing up to 500 berry plants and up to 200 virgin juniper plants during the year. This method showed the highest profitability of 1,333%. The vegetative method of propagation of berry was 130%. The profitability of the seed method was 0%. We have developed our own technology for growing berry and virgin juniper seedling under mother plants. The method of propagation by cutting of berry with the help of root formation regulator, cornamine, heteroxin, etc., can be considered effective and promising. Berry seeds quickly germinated in artificial conditions on the soil from under the berry mother plants, thanks to mycorrhiza forming fungi and other factors. Berry extract is in concentration of 20-30 gram per 100 milliliter in June-July can be used to create a herbicidal preparation. Compared to the control, berry extract inhibited the roots of Saxa radish by 600%. This was confirmed by many experiments in which berry completely inhibited the growth of roots of the Saxa radish. Also in experiments comparing berry extracts and the Roundup herbicidal drug, berry showed the same effectiveness as the herbicidal drug. Himalayan pine extract in concentration of 10 gram per 250 milligrams and in a dilution of 1 to 10 stimulated the growth of the roots of wet seeds of the Luzanivska variety and radish of the Saxa variety in June and August. The extracts of cedar of Lebanon and cedar of Atlas stimulated the growth of roots of Luzanivska wet in August, October, November, and December. Compared to the control, the extract of cedar of Lebanon stimulated the growth of wet roots in August by 667%. Compared to the control, the Atlas cedar extract stimulated the growth of wet roots in October-November by 213%. Leaf extract of cedar of Lebanon and black pine in a concentration of 50 gram per 10 liter were successfully used for root and foliar feeding of conifers. Thank you for your attention.